Hello and welcome to Genetics Day 3. So today's aim is what is DNA composed of and how does it replicate? And we're going to be talking primarily about the composition or structure of DNA, which hopefully you remember. It is made of nucleotides with base pairs, which we've already talked about, and it has a double helix structure, which we're going to be reading all about below. And its replication, we haven't talked too much about yet, but we're going to introduce that topic in today's notes. So let's go ahead and get started. Question 1. The molecule represented below is found in living things. Which statement describes one characteristic of this molecule? So is it the template for replication of genetic information? Is it organic catalysts are made up of these molecules? And hopefully we remember that organic catalysts, enzymes, are made up of amino acids because they are proteins. So it is not that. It is different in each cell of an organism. Well, we know that's not the case, right? All of our cells have the same DNA. Or cell membranes contain many of these molecules. And no, those would be phospholipids. And although DNA has a sugar phosphate backbone, it is not a phospholipid. It is the template for the replication of genetic information. So the answer to number 1 is A. All right, question number 2. The diagram below represents a section of a molecule that carries genetic information. The pattern of numbers represents a sequence of paired bases, the order of proteins in a gene, folds of an amino acid, or positions of gene mutations. And we can see here the numbers are repeating, and it's using 1, 2, 3, and 4. So hold on, let's color code that a little bit. And the base pairing rule is usually represented by letters A, T, G, and C. But by using these 4 repeating numbers, this demonstrates the same thing, right? It demonstrates the sequence of paired bases. We can see all the 1s are paired with 2s, and all the 3s are paired with 4s. Just like all the A's are paired with T's, and all the G's are paired with C's. And we can go ahead and, we missed one red one. But we're gonna see this sort of repeating color structure a lot today as we're talking about base pairing. Look at that beautiful strand of DNA. And we call one of these is the leading strand and the other is the complementary strand. And as DNA replicates, the leading strand serves as the template for the complementary strand. And this topic we're going to talk about quite a bit in living environment, but as you take more advanced biology classes, oh boy, this topic can get a lot more complicated. So, all right. DNA structure notes, part A. DNA consists of 2 molecules that are arranged into a ladder-like structure called a double helix. A molecule of DNA is made up of millions of tiny subunits called nucleotides. And nucleotides are made up of nitrogenous bases, which hopefully we remember from our biomolecules unit, right? Nitrogenous bases. And the nitrogenous bases have the same names as the nucleotides. So we have adenine, thymine, guanine, and cytosine. So each nucleotide consists of not only that nitrogenous base, but also a sugar and a phosphate group. And the nitrogenous bases are what get paired up in the center, and then the sugar and the phosphate group are what make up the backbone, as we refer to it, of the DNA. So this part that I'm shading in purple, this is the sugar phosphate backbone of the double helix structure of DNA. Look at that, beautiful, beautiful molecule. All right, so each one consists of the nitrogenous base, the sugar, which in this case is deoxyribose, and the phosphate group. And the sugar and the phosphate group make up the backbone. And the nitrogenous bases are AT, G, and C. Remember, all teachers go crazy. So cytosine and guanine pair together, and adenine and thymine pair together. The phosphate and sugar form the backbone of the DNA molecule, whereas the bases form the stairs, right, of the ladder. What are the four bases and how does the base pair rule apply to them? So, all right, we just went through those four bases. Let's go through it again. We have adenine, thymine, and that's what the base pair rule means, is that adenine and thymine always pair together. And then we have guanine and cytosine. And these words are a mouthful. So if you are like, I don't know how to pronounce that, just make sure that you remember the letter patterns, right? So A pairs with T, G pairs with C. Now, what is a codon? So a codon is a group of 3 nucleotides that code for a specific protein. And when we actually do our next state mandated lab, although you will read about codons in our next DNA structure lab, but when we do our next state mandated lab, you'll get the universal codon chart. So this is a sequence of 3 nucleotides, so like ATG, and then what does ATG code for? Which amino acid does ATG code for? But once we use the universal codon chart, you guys are going to have another one of these letters, uracil, which is only present in RNA. So a little sneak preview that is going to be coming up. But all right, now let's read a little bit about DNA replication. So we can see here we have the double-stranded DNA molecule, and then the strands get unzipped, they separate, and then based on those 2 template strands, you can see bases getting added to complement the template strands, right? So let's say this here is A, T, T, G. On the other side, it's going to match it up with T, A, A, C. So this is why the base pair rule is so important, because this is how DNA replicates. So that double helix structure gets unzipped, split into 2 strands, and then each strand gets a new set of base pairs to make 2 new daughter DNA molecules. And each one, as you can see with the color coding, has half of the original DNA strand. And this is how DNA replicates in the simplest of terms. So all DNA is made of a sugar, a phosphate group, and one of the 4 nitrogen bases. New cells are formed when a cell divides into 2 daughter cells. For example, cell division in the lining of your mouth creates the new cells that replace the cells that are rubbed off whenever you chew food. Before a cell can divide, the cell must make a copy of all DNA in each chromosome. This process is called DNA replication. Why is DNA replication necessary before each cell division in mitosis? So we already know this. This is a little review from Unit 6. But DNA replication must happen before mitosis so that each new cell has identical DNA, right? The daughter cells that are made by mitosis are always going to be identical. If the DNA doesn't replicate, if all the DNA doesn't copy, then mitosis won't be able to happen because the cell needs the instructions for life. Remember, DNA is the instructions for life. So if you don't have the instructions, how are you going to live? All right. Next, the process of DNA replication is shown on the right. During DNA replication, the 2 DNA strands of the DNA helix are separated in each old strand the old strand is considered a template. Okay, sorry, that sentence is missing a word or something, but the 2 strands are separated and the 2 old strands serve as templates for the new DNA molecule. The nucleotides in the new strand are added one at a time, and we can see that process happening there in the diagram. The old strand is considered a template. The nucleotides in the new strand are added one at a time. Each new nucleotide is matched to a nucleotide in the old strand using the base pairing rules. And this is why these base pairing rules are so important. Don't forget ATGC. The base pairing rules are G always matches with C, GC, and T always matches with A, AT. I need to pull out that shirt. Remember, all teachers go crazy, and this is important. And we can see each one of these little 3 collections of 3, these are codons. And these codons code for specific amino acids. And so many critical structures in our bodies are made up of amino acids, so those are very important. So a codon is just a group of 3 nucleotides, 3 nucleotide bases. So DNA replication results in 2 new DNA molecules that are identical to the original DNA molecule. So we can see there's the 1 original strand, it gets unzipped into the 2 template strands, and then the new nucleotides are added one at a time based on the base pairing rule to create 2 new daughter DNA molecules. And then after that, that's a fire drill. After that, mitosis can happen. That was just a fire drill. Sorry about that. I know you guys aren't even aware that 5 minutes have just passed, but that was just a fire drill. Those of you in-person students will remember that fire drill. All right, so all teachers go crazy. DNA replication results in two new DNA molecules that are identical to the original DNA molecule. Thus, each of the new DNA molecules has the same genetic information as the original DNA molecule. DNA polymerase is an enzyme that helps to make new matching DNA strands by adding nucleotides one at a time and joining each new nucleotide to the previous nucleotide in the growing DNA strand. So each one at a time according to the base pairing rule. Each new nucleotide added by the enzyme DNA polymerase, right here in the text. Let's use a different color. Let's make that stand out. Matches the corresponding nucleotide in the old strand of DNA in accordance to the base pairing rules. So now we're going to replicate the following DNA sequence. The template strand, AAT, TTA, TCC, AGG, GCT, CGA, ACG, TGC, ATG, TAC, GCA, CGT, ATG, TAC, GCT, CGA. All right, all according to the base pairing rules. All teachers go crazy. Describe the function of DNA polymerase. DNA polymerase is an enzyme that helps make the new matching DNA strand by adding nucleotides one at a time and joining each new nucleotide to the previous nucleotide in the growing DNA strand. And that ACE at the end should already be telling us it is an enzyme that adds new nucleotides during DNA replication occur according to the base pairing rule. What is the backbone of DNA? What is it composed of and what is its function? So the backbone is the sides of the ladder. Made of sugar and phosphate. And it is the structure of the DNA to create the double helix shape. Which really maximizes the amount of genetic information that can be contained within the DNA molecule itself. All right, so that is all I have for you today, ladies and gentlemen. Today's aim was, what is DNA composed of and how does it replicate? And we talked all about DNA structure. Remember, DNA is made of a nitrogenous base, a sugar, and a phosphate group. And we talked a bit about DNA replication, which we're going to talk even more about in the coming days of this unit. But don't forget that DNA base pairs are paired according to the rule, all teachers go crazy, ATGC. All right, guys, that's everything. Have a super fantastic day, and I'll see you all tomorrow on Zoom. Bye, guys.